According to French philosopher Michel Serres in his 1998 work, The Five Senses, we hear by means of the skin and the feet. We hear with the cranial box, the abdomen, and the thorax. We hear by means of the muscles, nerves, and tendons. I am the resonance and the tone. I am altogether the mingling of the tone and its resonance. It seems that Serres was right, with the discovery of cellular sounds in sonocytology 6 years later. Sonocytology comes from the Latin word sonus, meaning sound, and the Greek word kudos, meaning vessel. Scientists in this field study the vibrations and sounds that cells emit as a result of their metabolic processes. At the center of this discovery was Jim Gimzewski, a scientist in the Department of Chemistry and Biochemistry at UCLA. Gimzewski had previously worked with nanotechnology and was originally interested in the movement of cardiomyocytes, or heart cells. However, the stem cell samples sent to him by his colleague in Sardinia were seized by U.S. Customs in the height of 9/11. He ended up borrowing yeast cultures from a nearby colleague to study. Yeast, scientific name Saccharomyces cerevisiae, is a unicellular fungus that was the first eukaryote with its genome fully sequenced. Due to its durable cell membrane, it was a fitting specimen for studying cellular vibrations. In 2004, Gimzewski worked with graduate student Andrew Pelling to test for yeast cell vibrations with an atomic force microscope, or AFM. The AFM was created in 1985 by IBM researchers Gert Binnig and Calvin Kate. It's a special instrument with a probe that can map nanometer-sized objects that normal light microscopes cannot capture. With its 10 nanometer silicon tip, it's 100,000 times smaller than the head of a pin. The rest of the microscope is made up of a beam holding the tip that rests on the surface of the specimen. Any hills or bumps on the surface are detected by the laser sensor and reflected in the electronic mapping. While the tip would usually be dragged across the specimen, Gimzewski kept it in one place on the yeast cell membrane to record periodic motions of the cell. What they found was the cells vibrated around 1,000 times per second, or a frequency of 1,000 hertz. Human ears can hear pitches from 20 to 20,000 hertz, so the cell noises are well within the auditory range. However, the amplitude, which was around 3 nanometers at 30 degrees Celsius, was too low to be heard. Gimzewski decided to increase the amplitude, or in other words, turn up the volume while preserving the frequency. The resulting notes were just above middle C, more specifically between C# and D. Here is a recording of the sounds emitted by the living cells. However, there was the concern that the sound waves recorded by the AFM were emitted by the surrounding machinery and not the cells. To test this, Gimzewski also recorded the motions of the cells in different temperatures and solutions. For example, he treated the cells with sodium azide to inhibit metabolic activity. In this case, the cells did not oscillate. As well, oscillation frequency increased with temperature, suggesting that the motion is due to biological activity. Lastly, a series of control experiments showed that the AFM likely did not contribute to the results because the machine's resonance was at a much higher frequency than the cells. Here is a collection of the sounds emitted by the cells that underwent temperature and solution changes. Putting alcohol on them resulted in high-pitched screaming, which was likely due to molecular pumps working feverishly to expel the irritants. Popular Science articles even described Gimzewski as the master of the cellular torture chamber. Gimzewski also looked at the diagnostic potentials of sonocytology, as cancerous cells metabolize faster and vibrate at a higher frequency than regular cells. He claimed that sonocytology was preferable to other methods of studying individual cells, as it is non-invasive and uses no fluorescent markings or dyes. However, testing mammalian cell membranes would require special AFM tips, since they are much more fragile than that of yeast cells. His incredible findings were later transformed into a museum exhibition in June 2004, titled The Dark Side of the Cell. It was a walk-in immersive audiovisual event directed by artists Ann Niemetz and Andrew Pelling. It was the first composition to use cell sonics and involved projecting microscopic images of cells and their sonograms onto structures inspired by the actual structure of the cells. Currently, researchers are still working on refining the original experiment and testing the future applications of their findings. Some suggest creating a library of sounds to allow faster diagnosis of diseases. Whatever innovations stem from these findings, it's important to remember that yeast cells and other living organisms already make noise. Our duty is to listen, whether it's with an AFM or simply staying quiet, in order to hear and understand their unique songs.